So the first thing that you have to do if you're going to do any kind of genetic screening, genetic therapy, is to actually test the DNA. So sequence it, see if the gene that you know can give rise to a certain disorder is present in the person. So again, you get kind of a different form of restriction map, a DNA fingerprint here, but this is looking for specifically the gene. Okay, so you can see the N here represents the normal genes, and you can see with the patients they're either missing certain segments of DNA or, well, multiple segments of DNA or just a couple here. So that determines the risk of either the person having a specific disease or the risk of passing on a genetic disorder. Okay, because as we get into more of genetics, the genetic side of biology, we'll talk about carriers, people who have a single piece of a genetic disorder, and they can pass it on to a child who will actually have the disorder if their partner also has a piece of that genetic disorder. So there is a risk of passing on a genetic disorder even if you don't present any symptoms of it. So it can also be used to detect specific genes or proteins. So for those, generally you'd be looking for a sequence versus the fragment size patterns that you get here. Okay? So it can also detect some genes that are related to an increased risk of cancer. So not all cancers have a genetic component, but some of them do. And if you have a family that has a lot of instances of a particular disease that's not always genetically linked, it's pretty safe to say at that point that there is a genetic component to it. So you can also detect genes known to cause genetic disorders, like the gene for cystic fibrosis. So cystic fibrosis is basically an ion imbalance inside or across several membranes inside the body. It basically causes a very thick mucus to build up in certain regions of the body, including in the lungs and also in the pancreatal duct. So basically the little tube between the pancreas and the intestines. So because of all of this mucus buildup, they have a lot of trouble breathing at times. They have to break up the mucus in their lungs a lot. It's a very good environment for different lung diseases like pneumonia. And then they also have to take enzyme supplements because the pancreas that creates these enzymes can't actually get them out into the digestive system. So there is, well, the most common form of cystic fibrosis is caused by a single mutation in a gene, and so they can actually detect that specific mutated gene. So gene therapy is still relatively new. I think they have at least one form of it is in clinical trials right now, but it involves the replacement of faulty genes. So this would be a lot easier to do when it's still an embryo. Fewer cells, a lot less DNA to try to correct. But oftentimes you don't know that there is any kind of genetic disorder until the person is older. So that has been kind of the major issue of trying to modify the genes after the person has already started growing up. Okay, because then you have a massive amount of DNA that you have to try to change. So gene therapy replaces defective or missing genes. It can also add in new genes, specifically to treat a certain disease. Again, have to be very careful with this because you have to be able to add it into a very specific region of the DNA in order to not add it in the middle of another gene and disrupt the function of that gene. all different kinds of things like that. So it's quite a problem. It's a very daunting problem. There's a lot of data that has to be considered when ever doing any kind of gene therapy. So several experimental techniques are used for gene therapy. Again, most of this has still been done in lab animals of different kinds. It's not really all that approved for human use yet, though there was a recent news story about kind of illicit gene therapies that cause a bunch of issues. So I'll see if I can find that news story for you. So genetically engineered viruses can actually be used to infect a patient's cells. If you've seen I Am Legend, that is actually what they were talking about at the beginning of the movie. Of course, that went horribly, horribly wrong, but anyway. So in this case, it's kind of hijacking viruses' natural tendencies to infect cells, except in this case, you're delivering a useful gene into the patient's cells instead of the viral DNA. And then you can also insert a gene to stimulate the immune system, okay, which would help it target and attack cancer cells. Again, this is kind of an iffy sort of thing, because if you overstimulate the immune system, how's it going to tell between a cancer cell and a healthy cell and not attack the healthy cell and things like that? You can also insert suicide genes into cancer cells. So these are genes that cause the cancer cells to go through apoptosis or programmed cell death, or they will actually activate a drug. So the drug will only interact with cells that have these genes in them. But again, you have to think about, okay, what happens if the gene gets implanted into a cell that isn't a cancer cell? What happens then? How do you prevent this whole thing from just going completely crazy? So CRISPR, which was that video that I showed you back at the beginning of this semester, has been a huge, huge discovery right in the past just couple years, not even a decade yet. But it is DNA combined with proteins that can very specifically edit DNA. It's actually, it's incredibly precise and very accurate. So this has been kind of a big push in research, is looking at CRISPR. So gene therapy has many technical challenges, inserting genes into the correct cells and only those cells so that you're not infecting or harming healthy cells, controlling the gene expression, so how the gene is actually used once it's inserted, because inserting it is only half the battle. You have to actually be able to use it after it's in there. And then also determining the effect on other genes. So again, you have to be careful that it's not interfering with the function of other genes that are also very important for the cell. And then also when the protein gets produced from this gene, is it going to negatively affect other parts of the cell? So that is it for this chapter, actually. So there you go.